Okay, I think we can get started. Hello, everyone, and thank you for being here today. This is our first departmental seminar of the academic year, and very glad that at least some of us can be here in person together. It is my greatest pleasure and honor to introduce our speaker, Professor Wong, here today. Not only because he's a world-renowned statistician, for which you can find his achievements and the numerous awards on his CV, but to me, the most important thing is that he's my PhD advisor. Ever since I came to Michigan, I've dreamed of this moment happening one day, and here we are. The first time I met Wing was in the summer of 2006. It was the end of the second year of my PhD. And for some reason, I was looking for a new PhD advisor. One of my friends told me that Wing has recently moved from Harvard to Stanford at that time. So I sent Wing an email. Soon, he replied and set up a meeting with me. At that time, I was mostly worried about my lack of background in statistics. My PhD was in computational math, and I've only taken a few probability courses. Surprisingly, the meeting went well. And at the end of the meeting, Wing gave me a book to read, which was not a statistics book, but a biology book, Molecular Biology of the Cell by Bruce Alpers, if I remember correctly. When I joined Wing's group later, I understood why. I was by no means the outlier in the group. There were also students from computer science, electrical engineering, biology, etc. Some of them were developing algorithms, some building hardware, some conducting biological experiments. We learned from each other and worked together. Such kind of PhD experience was invaluable to me. That's what struck me the most about Wing. While I teach in class and tell students that statisticians need to work closely with scientists, Wing is being a real scientist himself. Wing is also a great mentor to me. When I graduated in 2009, the job market was terrible due to the collapse of the financial market. Wing encouraged and convinced me to stay as a postdoc. And 2 years later, I got an offer from this department, for which I deeply appreciate our senior colleagues here. But simply put, I won't be here today without Wing. So without further ado, I'm delighted that we have Wing here today. Let's give a round of applause to welcome him. Thank you.
Let me first share my screen. Okay. So thank you, Wei, for that very nice introduction. I also want to thank the department for giving me a chance to visit, although it is a remote visit, but I have enjoyed it so far very much, and I learned a lot from talking to your students and faculties. So thank you for the invitation. So today, I want to report to you some progress we have made on using an approach called Roundtrip for unsupervised statistical learning. And so in unsupervised analysis, you are given a sample of multivariate data points, and the dimension of the data domain can be quite large. And the goal in this analysis is to learn something about the multivariate distribution. Right? For example, in today's talk, we are going to focus on two aspects of the distribution. One is, if you're interested in estimating the density of the distribution, this approach can be useful for that. And also, we may want to ask whether there is any tendency for a data point to cluster, and if so, what are the clusters? And this approach can also be used for that purpose. So these are two examples I would give today. So let's start with density estimation. So a simple way to try to get a density estimate is to learn a transformation from a known distribution, right? So let's say if C has a known distribution, so the density of the variable C is known, and you seek a transformation G that would transform it to the data domain. and in such a way that it approximates the underlying distribution for X. And if you're successful in learning this transformation G, then by the transformation formula for density, you can calculate the Jacobian of the transformation, and then you multiply to the known density for C, and you get your density for X, right? So by estimating G, you are basically estimating the density. So this approach has been used recently in connection with a neural network to get density estimates. So these are the so-called neural density estimates. There are quite a bit of attention in this field. And their idea, in the existing work, is to try to get the transformation G by building it up as a composition of a series of simple transformations. And you want each of the simple transformation to have a Jacobian that is easy to evaluate or compute. And then you can get the Jacobian of the whole transformation, right? So the simple transformation are usually learned by deep neural network. You use a deep neural network to model this simple transformation. Now, this approach has some limitation because you have to do it this way. You are limiting the class of function G that can be represented through this manner. So our thinking is that, well, we may actually do away with the Jacobian requirement and try to directly learn a pair of transformation that would transform the data domain to a latent domain, where the latent variable C has a distribution which is either known or known up to a few parameters, right? And you learn the transformation H that go from X to C, and you also learn transformation G that go from C to X. And so on the one hand, you want the data generated by the latent variable going through the transformation G, you want that to have a distribution that is close to the empirical distribution that you observe in the data space, right? So C has a known distribution, so that would put a constraint on your G, right? But in addition, we also want the reconstruction error to be small. So this means that if you go through a round trip, you take a data point in X, go through the transformation H that takes you to the latent space, and then go back to the data domain using the G transformation. So you have a displacement from the original data point to a new data point. So you want that displacement to be small. So this we call the round trip error or the reconstruction error. You also want this to be small. So one requirement is to match the distribution. The other requirement is to have a small round trip error. And similarly, if you can do the same for the transformation C, right? You want to map X to C space, and you want to match the known distribution of C, and you also want the round trip error to be small. So if we can do this, then we have a way to estimate the density of X. Once we know this pair of transformation, I will explain how to do that. But one important point is that in this formulation, the latent space can have smaller dimension. Typically, we want it to have quite much smaller dimension than the original data space. So there is a dimension reduction property of this approach. Whereas if you insist on the invertible transformation as in the previous slide, then the latent dimension has to be the same. as the data dimension, right? Because it has to be an invertible transformation. But here we don't insist on invertibility. Okay, so let's see how this works for density estimation, and later on I'll explain how it works for clustering. So let us assume that C has a known distribution, say a standard normal in m-dimensional space, and X tilde is the mapping, from C to the data domain. Then our model basically is that the data is generated by latent variable going through this generator function, and then plus some normally distributed noise. So this is our underlying model for our density estimation. So then the target density in this model is very simple to write down, because given C, X tilde is fixed, right? So then you have X is normally distributed with X tilde as the mean, is a normal distribution. So this conditional distribution of X given the latent variable is just a Gaussian variable, right? But of course, Z is not known. So you have to integrate out C to get the margin of X. Oh, sorry. So this will give you this expression for the marginal density for X. So you combine the normal distribution of C to the normal distribution of X, you get a quadratic form in this exponent here, right? And then you integrate out C. So this comes from the latent variable distribution, which is assumed to be standard Gaussian. And this come from this model here. Right, so we have a closed form expression for the density of X, and we are going to use this as the basis for the density estimation. So before I talk about how to learn this function G and H, and then how to use it to evaluate this expression. Suppose we can actually implement this and get a numerical evaluation of this integral, at least approximately. Let's see how it works, right? So I'm going to show you some results first before we go into the method, the algorithm. So here's some simulation result. The true density, this is a two-dimensional visualization. So we have 3 true density, and these are several competing methods. The round-trip method is what I'm going to talk about, and you can see that it actually recapture the unknown distribution quite well. The structure of it is pretty consistent, right? Whereas these are 3 different neural density estimate built from those simple transformation using neural networks. I believe this method, MADE and MAF, both originate from Francis Bach's group. And this is work from Google Brain. So you can see that they capture some aspect of the underlying distribution, but there are quite a bit of distortions. And the distortion is especially bad for this method and for this involute function. You're not really getting it right. All right. So this example just shows you that the representation power of this simple neural network neural density estimate, they are not rich enough to actually represent this kind of distribution. The round trip seem to have more richer representation power. All right, so let's go back to the problem of how to learn G and H. So we're going to use adversarial training and to learn this function. So the function G is represented as a 10-layer artificial neural net. This is a dense network. And then the H is also a neural network, right? A deep neural network, 10 layer. But to learn this G and H function, we have adversarial training. So that will require you to have a discriminator that would try to discriminate the data point generated by mapping latent variables through G to data space and try to discriminate that distribution from the empirical distribution, right? So then the objective is that you train your G, given the discriminator, you train your G to make it hard for the discriminator to tell the data points apart. And given G, you want to train your discriminator network, which is a 4-layer neural network. You try to train that network to try to distinguish the generated data point from the actually observed data point, right? And conversely, you can also do the same for the H function, right? So you have 2 discriminator and 2 generator, depending on which direction you want to go. So this is the way we train this. Then we can just use a stochastic gradient to train these networks, and there are good software that you can use to do that. So, so after we have done with the training, then we know the function G and H, right? So let's now look at the problem of evaluating this expression. So we know that the density that we want to estimate is expressed as a mixture of normal density, where the mixing is mixing through this latent variable C. So you get this integral that you need to evaluate. Now, since this is an expectation, if you can generate sample from the latent space, which we know how to do because this is just a standard formal, right? Then you can just average this. Once you have C, then you have this normal density, right, for X. So the point X is given, right? So you can evaluate that normal density, and then you can now average out this, according to IID samples of C, right? So this is a standard Monte Carlo evaluation of an expectation, right? Now, but this naive way of doing it is very inefficient. The reason is that, so in doing it this way, you basically only need the function H, because this normal density depends on the mean is H of C, right? So you map C to, is G of C, yeah. G of C will give you the mean of this density, and so you don't need the inverse transformation H to evaluate this if you use IID sampling of C. Now, but this is very inefficient because most of the value of C that you draw will be if you evaluate it, it will be far from the observed data points, so that you're not putting the sampling at the right places, right? So you are sampling according to this part of the space or of the cost function, but you're ignoring this part. So we want to take this into account. So the way you can do that is that, given a particular X, you ask what are the C value that are likely to make this distance small, right? Because if this distance is small, then the density will be high, right? So of course, the way to do it is you look at the inverse mapping, because the inverse mapping would take a data point X to the latent space, and then you know that data point C, if you take it back to the X space, it will have a small displacement. So this is exactly what you need. So what you want to do is you take the inverse transform H of X and use it as a center for a T distribution with a suitably low degree of freedom, so that its tail is thicker than the normal. And this would be a very good proposal density for important sampling. So what you do is you draw IID samples from this density which has its mean determined by the inverse transformation from that X to the C space. And then you draw from this T density. And since this T density is not really a density you want, then you compute an importance weight, which is the ratio of the correct density to this proposal density, right? You know that the actual density for C should be this known density. Right? So this importance weight is computed, and then you can just do the weighted sampling of this conditional density of X given C. These are just normal density. So this expression you can evaluate, right? And this will give you an importance sampling based estimator for this interval. So this is why we need to learn a pair of functions. So G would map from latent space to X, and then X will map from data space back to C. And that will give us the value of C that we should focus on. Okay. So is there any question about this strategy? Okay. So let's look at some test results. So here we have 5 data sets that we took from the UC Irvine data set, a database from various fields, right? Some are from social science, from chemistry, and from finance, of various data dimensions, from 6 to 90. And we are going to use a latent space of lower dimension. Okay? So it's given here. And training sample size, validation sample size, and test sample size. So if you look at the various density estimate that we talk about, our method is called Roundtrip. And then there is several neural density estimate that has been proposed. And then finally, we throw in the kernel density estimate as a baseline comparison. Now, depending on the data set, the method, sometimes perform well, sometimes perform not so well, especially kernel density estimate perform very badly in some situations. Some of the neural density estimates are a little bit better, but you can see that the round trip is always quite a bit better. Now, better by what metric? We basically use the log density on the test data points, right? If you have a good density estimate, then the density should take higher value at the test data points, right? The test data points are not used to estimate density. So this is the metric we're using. So you're getting higher density values, right, on the test set using this method. Right, so these are different data sets. Now we can also use this method to generate data to simulate images, for example, and we can attach a quality score to these images. So here are 2 image data set, right? The famous handwritten digit data set, MNIST. So here the dimension is 784 for this image, right? And the reduced dimension in the latent space we take it to be 100. And the C5 images are lateral images, and they are higher dimension. So this is a 3000 dimension. We also reduce it to 100 dimension. So these are training data size, validation data size, test data size. So after we train our functions, we can sample from latent space and then plug into the G function to generate images, right? So we learned each digit is learned separately, right? So we learned the 0, how to generate 0 from this data set. And we are going to order the generated image according to the density value. The highest density value on the left side, the lowest density value on the right side. So you would bin them according to 10 bins of density values. And so the left columns are then the high quality image because they have high density, right? low density images are low quality images here. And you see indeed that the density quality score correlate quite well with the quality of the images generated. So this is the best of the three neural density estimate in the comparison, and we just take it, and you can see that first of all, the images generated doesn't seem to correlate as well with the quality score, right, that they generate. So I think, for example, this is not a good image for the digit 4, but somehow is regarded as higher quality. Right, so this is one interesting application, is to generate data and attach quality score. So this is the test on this CIFAR data set, and so these are images. These are real images, some examples of it. These are some examples of the images generated by our Roundtrip generator. And this is this competing method. So you can see that the images generated by the neural density estimate generator is not very good. You basically cannot tell that these are airplanes, these are cars, and you cannot see that these are really hard to see that these are both. But at least for the higher quality images generated by the Roundtrip model, which are on the left side, you can sort of tell that these are airplanes and these are birds, and the boats are also very clear. The automobiles are also okay. Some of the classes are not so great. I think, for example, the toes are very badly learned. Okay, so this is just some comparison result on image data set. So again, this image data set are pretty high dimension. So these CIFAR images has dimension more than 3,000. We also apply this approach to outlier detection. So the idea is that if you have a data set, let's say this data set, which has in 9-dimensional space, and we have a lot of training, validation, and test data. So we can learn the density, right, from this training data set. And after we learn the density, we evaluate the density for all the data points. And the data point that has particularly low density value, we can regard them as likely to be outlier, right? Outlier by definition should have low density according to the main distribution. So we use this density as a way to detect outlier. So we test this method on 3 data sets, again, taking from some database of outlier detection problems of various dimensions and various sample sizes. Now, the important thing is that the percentage of outlier within this data set, they can vary quite a bit. So we compare our method to the neural net neural density estimate, and also to 2 methods that are specifically custom designed for outlier detection. So this one is based on some version of SVM. This is probably based on some kind of random forest type algorithm. And again, the Roundtrip, so the metric for performance is the so-called precision at K. So that means if you know that there are K outliers in that data set, then after the ranking, according to the density, you take the K data points that are most likely to be outlier according to your density estimate, right? And then you ask how many of them are actual outliers. And so that is this index here, is the precision at K. So for the subtle data set, it's an easy data set. So everyone is getting pretty high performance, but still Roundtrip has a clear advantage, right? This method is not so great for this data set. For the mammography method, the SVM-based method is particularly bad. And then the neural network-based estimates are all performing better, but still, Roundtrip is tied with iForest as the best method. For this last data set, which is more challenging because the percentage of outlier is very small, all these methods are performing quite badly. But surprisingly, the SVM-based method now is better, but it's still not as good as the Roundtrip method. So again, this method seems to be offering something over the existing methods. Okay, so at this time, I think I can stop and see, is there any question on the first part of the talk, which is about density estimate, before we switch to single cell analysis? Okay.
Wait,
Yeah. Okay.
When you have, this is Laura, when you decide how many dimensions to reduce to, like in your top example, you go from 9 to 3.
Right.
If you allowed it to have more dimensions, I'm assuming in any one of these models, you would do better at some of these metrics, or how do I think about the trade-off between dimensions and ability to predictive ability?
Yeah, I think there is some implicit assumption that the data density is kind of approximated by distribution that is constant, that's near a low dimensional manifold, right? And there's a distribution on that low dimensional manifold that is generated from the G mapping. So this assumption, if it is satisfied in a particular data set, is a good assumption for a low dimensional embedding, then it is better to use that low dimension, right? So it just depends on actually the actual problem. If that assumption is not satisfied, then you are basically assuming that density is spread out smoothly over the whole space. In that case, I think this approach can still be used, right? This is now the embedding space has the same dimension as the data space, but I think the advantage would not be very dramatic. Yeah. So I think this is a problem-dependent question. You probably have to do some exploration by changing the embedding dimension to see whether there's any improvement, right? By increasing it.
Thanks.
Okay. Okay. Let me.
So we have a question from the students in the class.
I'll try to use
Okay. Can you use this?
Oh.
Yeah.
It's okay. I'll just try to yell. I am a little. I just want to make sure that I'm interpreting what you said about outliers correctly. So basically, when you said the precision at K, you're implying that you set K as the number of outliers.
Yeah, I cannot hear you so well. Can you speak?
So the question is about
the
question is about the metric of the K in terms of outliers. So you said there are K outliers regardless. Is that the question?
Yeah. So
yeah.
The way you determine K, so is this specific?
Well, so these are just evaluation of the comparison of the different methods. So in this data set, we actually know how many outliers are there. We know what are the outliers. So this is just a test data set. So for this data set, we know that, but in the estimation, we do not use the knowledge of the outlier, but in the evaluation, we use the knowledge of which data point are actually the outlier.
I see
and,
the
yeah.
number that you get, like the ones that you have.
So we just use the K is set to be the actual number of outlier in that data set.
Right. Yeah, okay. That answers my
Okay.
For the number, is that just the proportion of the correctly identified outliers?
So this is the, in this data set, there are 0.9%.
I guess the question is about the precision. What does the precision exactly mean? Is that right?
Well, so yeah, so you pick the K data points that are most likely to be outlier because they have the lowest probability, right? Then you ask what is the percentage of this K that are actual outliers? This is the precision at K, right?
Okay. So basically what it means is that for the forest cover, you have identified essentially a lot of false discoveries with your outliers.
Right, yeah. So that means you only identify, you make so many calls, but only 17.7% of those are actual outliers, right?
Thank you.
Thank you.
Okay. All right, so now turning to clustering analysis, I want to use single cell genomic as a background for this clustering discussion, because in single cell analysis, I think in this department, probably I do not have to go through the background of single cell genomic, but what you have is, let's say you want to analyze gene expression, then using various technology, and nowadays it's mostly 10x, you can easily generate a data set of, let's say, 10,000 cells. For each cell, you have a gene expression profile, right? Measured for that cell. Now, this data, of course, is rather sparse and in high dimension, and there are some dropouts and so on. So there are some technique that you need to handle this. But the the data set that we want to use is single cell attack sheet. So this measure chromatin accessibility of regulatory elements, and typically in a data set you get maybe 200,000 open regions, and then for each cell, the region can be classified as either open or not open, right? So this is the type of data that we want to analyze. A very high dimension, but also have lots of data points. And we are interested in doing cluster analysis. Clustering analysis basically asks, well, from the accessibility profile, can you group the cells into different clusters that are well separated from each other? All right. So the standard way of doing this is basically you pre-process the data by calling peaks, and then maybe doing some thresholding to call open and close, and then you do some kind of dimension reduction, right? You can use PCA if you want, or matrix factorization, and then after you reduce dimension this way, you would then visualize the data point, or you do clustering on the embedding space. So the clustering is done separately from the embedding, and usually the embedding is a linear embedding. Although lately people have been starting to use variational autoencoder or game models to try to generate the embedding. But still, you do embedding first, and then you do clustering on the embedding space. So we're asking whether we can sort of integrate everything together using, again, this Roundtrip framework. So, so here, we still have the Roundtrip model, just like before, with a pair of functions. But in the latent space, we have a continuous variable, Z, which typically will still be taken as a multivariate Gaussian. But then we have a set of categorical variable, let's call it C. So in the simplest setting, C is just a one-hot encoding of class labels. So suppose you assume that there are 10 clusters in this data. So it's just like doing k-mean analysis. You have to try various k, right? So let's say k is equal to 5. Then you have 5 cluster. Then this C vector would be 5 dimensional, and with a 1 hot encoding of the cluster. So if cluster 1 will have a 1 here and 0, 0, 0 for the rest, right? So then you concatenate this C and the categorical variable into the latent representation of the data point. So then if you learn the G and H mapping, once you learn the H mapping, then the observed data point can be mapped to this latent space. And once it's in the latent space, the categorical variable can tell you which cluster it belongs to. So you're doing clustering together with learning of the functions. And the learning is still done by this adversarial learning through the discriminator, dx and dc, right? So once you have the latent space distribution learned, right? So then you can do various analysis, like you can now cluster them according to this 1 hot encoding, or you can visualize them by using this vector in, plug it into t-SNE and then visualize that, right? Or you can do some trajectory analysis, which I will explain later, right? So one important feature is that now the latent distribution is no longer completely fixed. The distribution of C is completely given, but the distribution of the categorical variable would depend on a parameter, which basically gives you the relative size of the different clusters. And that has to be learned together with learning the G and H function. So those will be updated as you learn the G and H functions. You will also update the class weight, just like in k-mean, you have to reassign to the clusters. Okay, so here's some details of the loss function. There are some loss functions that are due to the adversarial training, like the generated adversarial network cost function, and then there's the round-trip reconstruction error. But here, in the latent space, you have some categorical variable. So for those, your error will be measured by cost entropy instead of the Euclidean distance error. So the total loss will be the sum total of all this. And training is as usual by iterating between learning the generator functions G and H and the discriminator function dx and dc. So given one, you update the other, and then you iterate. And then the learning is using this stochastic gradient descent type algorithm implemented by the Adam optimizer. And all networks are fully connected, except when you apply it to image data, then you have to go through some convolution layer, beforehand. So here are some data set that we use for evaluation that we collected from the literature, and they have various number of cells in the data set, from 1,000 to 80,000, and then the number of peaks score, and for 4 of this data set, they have ground truth label for the cell types. So the 6 cell types in this simulated data set, 8 cell types in the brain data, and so on. Now, but for the mouse atlas data, they don't actually have ground truth label, but the team has actually manually go through a lot of trouble to annotate their cluster. So they have 30 clusters, and they identify the most likely cell type So we are going to use that manually annotated data as the ground truth in the evaluation. So we want to compare our method to several other commonly used packages, right? So SBABC is a method that we published several years ago. Then Scale and SysTopic is a very popular method. And the different methods have different functionalities. Some are capable of only dimension reduction and not doing clustering, and some are doing clustering at the same time. Actually, none of them. We are doing clustering at the same time, and so on. But these are some of the more popular methods available at the time we did this work, which is like 18 months ago. Now there are more methods, I'm sure. Now, so of the 4 data set that has ground truth label, we evaluate the performance by several common index that people use to judge the quality of clustering results when compared to ground truth label, right? So these are things like adjusted rank index and mutual information and so on. And you can see that no matter which index you use, the SCDAT, which is our method based on the Roundtrip model. So SCDAT means single cell, deep embedding, and clustering, simultaneous clustering, right? So they always seem to be performing either the best or very close to the best, right? And as for the other methods, sometimes they do well in one data set and not so well in the other data set. SysTopic, I think, is doing quite decently most of the time, but it's still, clearly worse than our method in some situations. So any question with this evaluation? This is evaluation on real data with ground truth label on the clusters. And in all the method, we assume the number of cluster is given as the ground truth. So just to evaluate the performance of the clustering algorithm itself. And so using this method, we can now cluster the single cell, right? So for example, if we look at the 4-brain data set, we cluster them and we compare to the ground truths, seem to be doing pretty well. We also look at whether the clustering makes sense from the point of view of transcriptional regulation by transcription factors with known motifs. So there's this software called Chromeware, which we can use to compute the enrichment of motif binding for a given transcription factor. Let's say you take a transcription factor like LBX2, and you can look at each cell and compute an index of whether that transcription factor's motif, sequence motif that you recognize, is enriched in the peaks, the accessibility peaks in that cell, right? So then you can use this and look at how the cluster perform with regard to this motif enrichment. And you can see that they do cluster separately in terms of these transcriptionally important features according to transcription factor motif analysis. So these are the label obtained by our clustering and the corresponding to ground truth label, right? So we can see that some of the excitatory neurons are not well separated, not well aligned with the ground truth. There are some mistakes there, but by and large they are pretty consistent. And we also spend a lot of time looking at the individual transcription factors and see how they express in the single cells. And it also seemed to make a lot of sense, which neurospecific transcription factors are in fact expressed in the right clusters that we identify. An interesting application that we find is that we can use this method to generate more accurate imputation of missing data, right? So this is in the hematopoiesis differentiation data set, and we know from immunology that there is some differentiation pathway that go from hematopoietic stem cell all the way to different types of progenitors, right? So we're particularly interested in this pathway from MPP, which are the multipotent progenitors, to this lymphocyte specific progenitors, common lymphocyte progenitors, CLP, through this LMPP, so some intermediate step. So we assume that we only have, let's say, the MPP data and the CLP data, right? But we actually don't use the LMPP data in learning the GNH function. But then we learn that function, and then we can ask whether we can interpolate, right? So we have the categorical variable, which has a one in that in this position, and then the other categorical variable has a 1 in a different position. Now you can interpolate these two categorical variables by doing a convex combination of them, right? So if you have a cluster for the digit 5 and another cluster of image for digit 6, by changing the mixing proportion in this convex combination, you can transform one image to the other, right? So we can use the same idea. We can transform the accessibility profile of an MPP by changing this alpha value. We can change it to a CLP profile. But when you set alpha equal to 0.5, you're supposedly generating something that maybe is closer to the LMPP. So these three colors are indicating three values of alpha. This is 0.5. So when alpha is 0.5, your accessibility profile that are imputed using the MPP and CLP profile is indeed close to the observed profile for LMPP. Right? So this is in the embedding space. So here we're doing interpolation in the embedding space. Now, of course, you can do the interpolation in the original data, or you can do it in the reduced dimensions PCA space, right? Or you can do it in the reduced dimension latent space learned by our round-trip model. And you can see that the correlation of the learned pattern to the actually observed LMPP pattern, the correlation is higher using our method, higher than the PCA-based interpolation, which is again higher than the raw data interpolation. So this is kind of interesting to us that we can look at this kind of trajectory of the transformation. Now in this data set, we also have several donors. So some of the LMPPs are coming from one individual and some from the other. So there are two donors and 3 cell types. And you can actually look at the embedding space and visualize the features that. So the embedding space has 3 categorical variable here, and then there's the other 10 dimension are the continuous dimension for the C vector. So the categorical variable basically give you the 3 cell type, right? And then if you look into the continuous variable, some of them are actually reflecting the different in the donor effect, right? So the different donor give you that. And some other features, you know, are probably about the more detailed patterns within the cell types. So there's a lot of information to be gleaned from looking at the structure of the latent variable. So you can separate biological cell type effect for donor effect, and you can use this to maybe adjust for donor effect if you want to. And finally, very important to us is that this algorithm is very scalable, because this deep learning based optimization is very efficient. They have very good software of doing that, and stochastic gradient, you know, in each update, you only have to look at a small subset of the data set. So the learning is very efficient. So when we try to run the different methods on the largest data set, which has more than 80,000 data points, 4 of those methods, baseline method, basically fail to run. It doesn't produce result after a long time of running, right? But you know, our method still runs in decent time. The fastest method is Scale, but we're slightly faster than us. This topic also can be run, but we've already shown that our method actually, in terms of performance, is better than this. In terms of speed, it's sort of comparable. So it's a highly scalable kind of algorithm for learning. So the final point I want to make is that the framework actually is very general. So here I have demonstrated how to use it for density estimation and for clustering. But by playing around with the distribution of this variable that we use to augment the latent Gaussian variable, this variable can have various structure, right? So in the clustering, we use one-hot encoding as the structure. In some other analysis, for example, we are currently analyzing some time course data. This variable C can encode some time information, time and cluster information. And then once we learn it, then you have that. We have information about you know, time development and so on. So this is quite a flexible scheme. This is the reason why we favor this approach from something like variational autoencoder, which is also a generative model, which can be used to estimate density. But there, the latent space distribution is not very structured, because it's basically the empirical distribution after you map the observed data point into the embedding space. So you don't have that flexibility of doing this more sophisticated modeling in the latent space. So finally, I just want to acknowledge contribution by my co-authors in these 2 papers, one on density estimation, the other one on single cell genomic, which are published in earlier this year in these 2 different journals. And the most important collaborator is Professor Wei Zhang from Tsinghua University, and Qiao Liu is a student, is a postdoc in my lab, but he's a former student of Professor Zhang in Tsinghua. So the work started while he was still in Tsinghua. So I would end here and try to answer questions. Thank you.
Thank you, Wing. Let's give a round of applause to the speaker. So our new format is like we take a 5-minute break and then people come back so that
Okay.
you take a break.
Sure.
So we all come back in 5 minutes.
Yeah, I'll just stay on the line. So yeah.
Thank you.
Okay.
So everybody, you can take a break for 5 minutes, and then we have questions after that. Okay, I think we can now back to questions. So I have a question is that, so you use this independent Gaussian for the Z, right? Regardless how high the dimension is. You talk about example where Z is like a 100 dimension. You still use independent Gaussian?
Yeah.
So does that make any difference?
Because
I know
we were basically pushing all the learning to the function G and H.
I see. I know your G can transform Gaussian to any distribution needed, but the 10, do you see like a 10-layer network is enough?
Yeah, we haven't play around with that too much. We basically just fixed the architecture, and it seemed to perform okay in so far in our examples, right? So it's quite a variety of examples that we have tried.
Let's see.
But of course, we can we can explore that later when we have some bandwidth. But right now we just more or less fix the architecture and just apply it as a, you know, as a sort of a routine tool, yeah, rather than trying to adapt it for each application.
Yeah, another question I had is about this GPU, learning, right? You say it's pretty fast, but you list the machine there, probably dedicated for this task. But I also heard, in places like a Google, they really need a very, very powerful machine. Like every hour take $1 million to train things.
Right.
Do you see, why
our networks are nowhere near what they're using, right? So we don't have really that complicated a network, right? We only have 10 layers. Our dimension. So the way we do it is we still first do a linear dimension reduction, right? Either by PCA or non-negative matrix factorization to reduce it down to maybe 200 before we go into the neural network setting. Yeah, because the neural network that we have are dense network.
I see.
Yeah, for the image data set, then of course we go through the convolution layer and so on, right? So so there's some reduction there before you fit into the dense network. So the number of parameters are not really that huge. So we just have one GPU, as you can see. It's not, we don't actually need a lot of computation. All the computation is done with one GPU.
Any other question?
This is Zheng. I have two quick questions, hopefully. So the first question is about the generalization of the method to discrete observations. I wonder whether that's whether that's quite easy or quite difficult, and how to do that. That's kind of my curiosity. The second question is, in this.
I don't understand. So the in the data space, you are talking about
Right.
the data space as discrete.
Yeah.
Distribution. Yeah. Yeah, it should be doable, because I mean basically the clustering, if you can do the clustering problems well, then intrinsically you should be able to handle a discrete variable.
So essentially, are you suggesting still trying to use the Gaussian
Yeah.
distribution?
So the Gaussian, the latent space distribution is not that important, because through the G function, you can transform it to be very close to discrete. Right. Just like those clustering problem, right? We have a grid of points in two-dimensional space, right? Basically, they're like discrete points, right?
Right.
The question, yeah.
I
I think the one thing that I come to appreciate about the deep neural network is that because these functions are very flexible, so you can produce a lot of patterns by playing around with the function. Yeah.
Okay. Yeah, and the second question is about the dimension of Z there. So clearly, you need to specify this. I wonder, what's your experience trying to choose that embedding dimension?
Yeah, so far we actually have not sort of done much in getting a good general guideline of this problem. Basically, we just pick something that seemed to be appealing and then just use it. I think if your actual data set can be well approximated by a distribution near let's say a 10-dimensional manifold, but you use a 12-dimensional manifold or a 15-dimensional, that's not going to hurt you too much, right? You're sacrificing something. But if you use much lower than 10, then you may be in trouble, right? So probably, in real application, we may need to increase it and decrease it to see whether there's any change. But it's just like k-mean clustering. What would be a good automatic way of choosing k? That's kind of hard. Yeah. So yeah, that remains to be we haven't spent too much time on that.
Yeah, thanks. That's my questions. Okay.
Other question?
I'm expecting a lot of questions from students, especially. Students should be asking questions.
Yeah, I mean, I do want to ask a question, as a student, when I was a student working with you, I'm really amazed by the type of problem you pick to work on, which are often out of the box that I'm thinking of. And also, you work on many different problems. So how do you pick the problem to work on? I mean, I think that's the question, benefit everyone.
Well, I think first of all, you need to have some kind of long-term sort of overarching goal in your research program, right? So in my lab, it's all about gene regulations and how it relates to maybe precision medicine. And within that framework, then we have these various data types, right? Some single cell data, whole genome data, what have you. And then if I see there is a great need in method for handling some types of data, right, that are becoming abundant, and it has important bearing on the overarching scientific question that I'm interested in, then I would focus on those. Right? So this neural network kind of tools, we pick it up because the single cell data is really driving us into these methods. The more standard statistical methods that just doesn't seem to, they don't seem to perform as well in our hands. Whereas this newer type of method, first of all, they are very easy to use now because of all the software developed by the big companies that are made freely available. And so I guess it's driven by basically a good overarching scientific goal, and then it's up to your ability to identify, within that, what are the upcoming problems, right? I don't know whether that's a good answer. I never thought much about, how I pick problems myself. It usually just comes sort of by chance. But I guess in the background, there has to be a drift, right? If this, you're worrying about some important questions, then there's a force pushing you towards the more relevant questions that you want to spend time on.
Thank you, Wing. That's very helpful. I have a question from the student here. Yeah, come here. Come here. The microphone, doesn't work long distance.
Yeah, sorry about that. Hi, Professor Wong. So, first of all, I am really impressed by the model, especially at the end when you were talking about how it can adapt both like as a stationary model and like track things through time. I thought that was really cool because I like time-related data. But I do have a question that's probably, I'm not very familiar with deep neural networks, so I'm not sure if that's something related to that. But when you were talking about like outliers and stuff, I thought it was kind of strange that it kind like when you have very few outliers, it was having a hard time like trying to figure it out, like trying to identify it correctly. I thought it would be the other way around, but I'm not sure if it's something specific to the model that kind of caused that issue. Like, I'm just wondering if you can say a couple words about if that's something that's weird that we're expecting to see.
You're right. I mean, if there's small number of outliers that are very far away, right?
Yeah,
then
that's
you'll be able
true.
to get it. Yeah. The problem probably, we don't know what's the real reason for that problem. It could be that it's both a smaller number of outliers, and the outliers are not so well separated, right?
Okay, that's what I was
main part
thinking. I was
data,
like, maybe
yeah.
it's something to do with the outliers, like
Yeah,
how
that's
far out.
Yeah.
Okay, that makes sense.
Mhm.
And just like the overall interpretation of the model, I'm not sure if I was interpreting quite correctly. So when you're applying this Gaussian distribution, you're essentially fitting the data into like kind of like a Gaussian model, and then that's how you identify like the
I think that the way to visualize the model is that you imagine data points lying in a high dimensional space,
right.
let's say 100 dimensional space, but in such a way that they are all kind of close to a lower dimensional space, let's say a 10 dimensional manifold, right? So there's that manifold, but the data points are not uniformly spread out across near the vicinity of the manifold. Within the manifold, even if you project it onto the manifold, there is still a density.
Right.
And that density is generated by the G function. The G function basically map your latent variable into that manifold.
I see. And that's why you called it a mixture of model, model.
Yeah.
Okay, that makes sense. Thank you so much.
You're welcome.
Thank you.
Any other question? Yes, we have one more. Please come.
Wait.
Professor Wong, yeah, I have a really basic question. So I saw like you divided the data into the testing data set, the learning data set, and the validation data set. Like I saw a really specific number of that. Is there like a overall rule, like what percent of data you divided into those three different data sets?
Yeah, well, that's a good question, but in our setting, actually, that is not up to us. It's the UC Irvine database. When they provide this data, they already have test data, right, and validation data divided. I think.
Okay, so you mean like the data set already provide the different sets?
Yeah.
Okay. Do you have any advice, like if we get a new data set, like an enhanced data set, and we
typically the learning is through maybe some kind of tenfold cross validation, right? So something like that, and you basically leave out 10% as a validation data set to help you sort of fix hyperparameters, and then the test data set typically maybe another, depending. I think it depends on your test data set has to be large enough so that it gives you a good evaluation, but it doesn't have to be a fixed percentage of your overall data. But I think something like 10% seems like a good number to me. I don't know. I'm not really expert in the nitty-gritties of this machine learning. Maybe someone in the audience like G, maybe better question per person to answer that.
Yeah, thanks.
Any other question? Okay, I think really appreciate Wing being here today. Give us a fantastic talk. I know the time there is already past lunch quite a bit. Thank you for being here. Thank you all for being here.
Thank you all for attending and asking good questions. Thank you. Bye-bye.
Thank you. See you.